Welcome to Chapter 2. Chapter 2 is mainly the tools of the laboratory. So we're going to talk about all the different things that are used in order to identify organisms. So, and this is part 1 of Chapter 2. So we're going to start out kind of referencing the 5 I's of microbiology. And you have this picture in your book. The right side of this slide shows kind of the 5 I's and what specifically they are used for. And we'll go through this throughout this PowerPoint. So inoculation, incubation, isolation, inspection, and identification. And the last two, inspection and identification, are generally kind of lumped together. So it's not, you have to inspect it in order to identify it. So we will, when I talk about that, it's going to be more so together. So inoculation is when we essentially take a sample from a patient and apply it or insert it or inoculate some sort of media with that specimen. And then we take that inoculated specimen and we incubate it at a certain temperature, usually for samples. Most of the time, it's going to be at about 37 degrees Celsius that we're going to incubate that in a little cabinet because we're trying to identify, at least in the medical field, we're trying to identify, what might be causing a infection or a disease or whatever, and we want to incubate it at body temperature, and 37 degrees Celsius is body temperature. Then once we incubate it, usually for 24 hours, we'll look at it the next day and we will note if there is multiple different microbes on that plate growing. Some may be the normal flora from our skin or our body, depending on where the specimen was collected from. And we need to isolate what might be the pathogen within that culture. So we need to separate out or isolate the pathogen from the normal stuff that is part of us. And then once we are able to isolate, then we're going to inspect those isolated colonies on the plates and then also identify by what we see. And also sometimes we'll do additional testing to verify. So that's kind of just the quick summary of what we're going to talk about. So again, inoculation is to, we want to inoculate on a culture or some sort of medium. And so these are terminology. This is terminology here that you should know what these words are. So a culture is to grow microorganisms. A medium is the nutrients for the growth of microbes. So what we put it in or on. Inoculum is a small sample of the microbes. Inoculation is the introduction or to apply the inoculum into or onto the media. And we get specimens from body fluids, discharges, anatomical sites, diseased tissues, urine, stool, sputum, skin, wound, all sorts of places. I'll go back here, and blood and pleural fluid and cerebrospinal fluid and so on. There's pretty much anything that can come out of the body, we can culture it. So again, incubation is a temperature controlled chamber, or an incubator is a temperature controlled chamber, and that's how we incubate the specimens. And I said earlier, it's usually 37 degrees. I'd say, 95 out of 100 specimens are incubated at 37 degrees, but occasionally we have to incubate things at a different temperature if we suspect it might be a certain type of microbe, because some microbes actually have a broader range of growth. We also determine the atmospheric gases that are needed for any given culture. It could be oxygen, CO2, just depends on what we're testing for. And the incubation period, the microbes multiply and produce growth on that or in that media. And 9 times out of 10, 10 times out of 10, we can observe that growth macroscopically with the naked eye. So this is just examples of a couple of different things. There's pure cultures, mixed cultures, and contaminated cultures. So, pure culture is a single individual microbe growing in whatever media we put it in. And these are illustrations or images of that. And then this is a mixed culture where we take 2 cultures and apply them to a plate, 2 separate pure cultures and apply them to a plate. And you can see that we have some yellow colonies. These little circles are called colonies on a agar plate. And then mixed in there, kind of in a homogeneous way, are these kind of off-white, clearish colonies that are kind of mixed in there equally. So that would be referred to as a mixed culture. When we have a plate with many colonies of a certain type, and then all of a sudden there's just one blob of something else, or 1 or 2 blobs of something else that don't belong, it seems like that is an indication of a contaminated culture, and that would be not good in the lab. We never want to contaminate our cultures. We always want to make sure that they are only growing what we intend from the patient. So media can come in different physical states, different chemical compositions, and then there's different purposes for different medias depending on what might be added into them. And we're going to talk about that moving forward here. So 3 main physical states: liquid, solid, and semi-solid. As far as media goes, it can come in a liquid form, it can come in kind of a semi-solid, and then it can be a solid form. Agar is basically a complex polysaccharide. At room temperature, it is solid. It liquefies at 100 degrees Celsius. It's flexible and moldable, so we can pour it into Petri dishes and have it, take the shape of the Petri dish, or we can pour it into a test tube and then lay the test tubes kind of at an angle, and we can end up with a slant in that test tube of agar. And agar is not digestible by microorganisms, so most of the time anyway. So that means it's going to withstand the incubation and the microbes growing on it. So that's just an image of what we just talked about. So we have general purpose media. So general purpose is, kind of exactly how the name, explains it. It's going to grow a broad spectrum of microbes. It's not going to be picky about what it grows. It's going to grow everything. And in the lab, depending on what we're growing, we don't use a lot of general purpose media because we want to isolate things. We want to keep the normal flora from growing, or kind of tamp it down a little bit, so it's not overtaking whatever pathogen might be growing in there. So general purpose media is not something that we normally use all the time in the lab. We also have enriched media, and that contains certain things. They put additives in that, like blood or serum, hemoglobin. Sometimes they put growth factors, depending on the type of media. And we use enriched media specifically for fastidious microbes. And fastidious microbes basically implies that they're picky. The microbes, will grow better on these types of media because there's things that they prefer enriched in the media. So we use enriched media in the lab to encourage the growth of pathogens if they're present in low numbers. So, if we use an enriched media such as blood agar, and the plate is covered in this one particular microbe, then we know that person has a pretty good infection. But the enriched media will also show if the person has, an infection that isn't rampant yet. It will pick up on subtle pathogens. So you can see this is an example of blood agar here, and this is probably chocolate agar, and it doesn't have chocolate in it, by the way. It's actually boiled blood, and it changes the color to a chocolate color. So you can see here that this agar was all blood, or all red at one time, and then when we put the sample on here, it kind of the microbes kind of ate up what was around them. Now, it didn't eat the agar because we said that it's not consumable by microbes. That the solid agar, the gelatin that this enrichment is put into. But the stuff that was added to the agar is what the microbes took advantage of and ate up and consumed, and it causes the agar to go clear in its natural state. But we'll talk about that later. So selective media contains one or more agents that inhibit the growth of certain microbes. So if you're selective about who you hang out with, that's the same idea. You're only going to let some people in and keep your circle small. So selective media is important in the primary isolation of a specific type of microorganism from a sample that happens to contain dozens of different species. So perfect example, if you do a throat culture, your mouth is loaded with bacteria. Your throat has bacteria. So if we use a selective media, it is going to be selective in what it allows to grow. And using selective media actually speeds up the isolation of the pathogen by suppressing the unwanted background noise or background organisms and favoring the growth of the desired ones. And you're going to have to know the different types of media and what their purpose is, and more stuff as we cover here. Differential media. This allows for multiple types of organisms to grow, but they look different. It displays visible differences in how they grow. And differential media will show variations in colony size or color. There'll be media color changes. So just like I showed you earlier, that plate changed, the red went away. Same idea here. The media can have color changes, and that depends on, in some cases, pH, production of gas, and so on, will cause all these differences in how the media looks. And based on how the media looks, then we can narrow down what microbes might be growing on that particular media. So variations come from the chemicals in the media with which the microbes react. And this is an example of that. So we have the same mixed sample. We put it on general purpose media, and we put it on selective media here, and we put it on differential media here. So this is the same mixed sample, okay? Take this sample, put it here and here, and then we moved it over here and put it on here and here. In the general purpose media, it's all going to kind of look the same. It's hard to differentiate what the different microbes are. But on selective media, it's going to suppress all that normal flora, all the stuff that we don't care about, all the stuff that's not causing the infection. It's only going to allow, in most cases, the bad stuff, the pathogen. Okay. Differential medium, on the other hand, it will allow for many of these organisms to grow, but it will change color. It will display differences in personality, if you will, of that microbe on that plate. So we're able to distinguish between two plates. Maybe we're able to come to a specific culprit that is causing the infection. So I'm going to stop here, and I'll pick up on part 2, chapter 2, in the next recording.